Hello and welcome. Thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator here at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Rebecca Humphrey, Principal Data Manager, and Dr. Mitali Gaurav, Assistant Director of Pharmacometrics, will discuss data needed for the best analysis data set for PK/PD NLME modeling for multiple software and the importance of collecting meaningful data. The capture of appropriate dosing data will be the primary focus as it can present many challenges. Samitra, Account Executive for Business Development, will serve as our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously. By registering for and attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all questions in the Q&A after the presentation. Take it away, Sammy. It's all you.
Thank you, Jasmine. Hi, everyone. Good afternoon, good morning, good evening, based on where you are in the world. It's a little bit about me. My name is Soumitra Rothekar. You can call me Somi. And I work along with Rebecca and Mitali and Jasmine here, focusing on the client relationship in the business development function. And it's my pleasure to introduce two of my wonderful colleagues, Rebecca and Mitali. So first, Rebecca. Rebecca Humphrey is the principal data manager here in the clinical pharmacometrics, clinical pharmacology division here in Simulations Plus. And she has over 30 years of experience working with scientific data. She has been a part of Simulations Plus data management team for over 15 years. Her expertise is in building complex analysis datasets. She has contributed to a multitude of projects over the years and worked with a variety of data in all formats. Prior to pharmaceutical industry, Rebecca managed environmental data. A little bit about Mitali. Mitali Gaurav is PKPD scientist here at Simulations Plus. After completing her PharmD, she did a postdoctoral fellowship in pharmacometrics. Her expertise handling variety of data sets comes from having worked on multiple projects spanning non-compartmental analysis, population PKPD, exposure response modeling across diverse population cohorts, and therapeutic domains. So with that, I welcome Mitali and Rebecca for this particular webinar/presentation. And my request to all of the attendees is, kindly do put in your questions, your remarks, your comments on the chat section below. We'll do our best to address them towards the end of the webinar. And if we are not able to address them during this time, we'll be more than happy to follow up with you offline. With that, Rebecca and Mitali, kindly take it away.
Thank you, Somi, for the introduction. Hello, everyone. I am Mitali Gaurav, and today Rebecca and I will be discussing the importance of capturing and using meaningful data for population PKPD analysis with nonlinear mixed effects modeling. Before we dive in and discuss, next slide, please. Before we dive in and discuss in detail about the main agenda topic for today, I would like to spend some time discussing what is POP PKPD analysis dataset, where does this information come from, and why is it important for modeling. So first part is, where does this information come from which creates our data set? So usually the information comes from the clinical trials, and during the conduct of these clinical trials, a lot of information is collected. So when the time comes to perform the analysis, one should be very aware of the modeling goal, the end result that one is trying to achieve, and also what kind of data is necessary or required to achieve those goals. So usually what happens is these data sets which are intended for the analysis are the arrangement of the information which is collected during the clinical trials and are assembled in a way which is compatible by the software that one is going to use for the analysis. Generally, these data sets are time ordered, where each record represents the time course of events related to the drug administration. And if your analysis is about pharmacokinetics, then that will include your resulting concentration. And if you're dealing with pharmacodynamics, then there will be records pertaining to your pharmacodynamic outcomes that you are trying to analyze. Usually, as we add on the data in a row-wise fashion for each individual in these data set, this gives the longitudinal nature to the PKPD analysis data set, where each record represent different kind of events. It can be either your dosing, your pharmacokinetic sampling, or your pharmacodynamic responses. A point to keep in mind here is that this time ordered data sets need to happen prior to your analysis. Because there are some softwares, for example, Monolix, where you can put in your data in any order and it will arrange these data in time orderly fashion for you. So just keep in mind that you need to have your data set in time ordered fashion only prior to your analysis. You can put it in any fashion into your analysis software. Coming to the question now, why is capturing meaningful data essential for modeling? And that's because data is the most critical component part of your modeling. If you do not have the data or appropriate data, you will not be able to perform your analysis and get the results that you are aiming to achieve. For example, say if you're interested in analyzing or determining if creatinine clearance has an impact on clearance of your drug, then you need to make sure that you're capturing creatinine clearance in your analysis data set. The other example would be if you're trying to say characterize the PK of your drug in pediatric population, for example, then you need to make sure either you are enrolling pediatric population in your clinical trials, or you have a or a study already taken place that you want to include in your current data build, which includes pediatric in that particular therapeutic area. And that's because the data determines the modeling results. Whatever data you have, you will only be able to perform your analysis on that. But the other thing you need to keep in mind is that the data that you capture is also determined by your modeling goal. What is the end result that you are trying to achieve by doing or performing these analyses? Next slide, please. So, as you can imagine, when you have a lot of data to work with, creating a data set where you can perform these analysis can be a very tedious task, especially when you have different scenarios to deal with. So for example, example of complex data can be when you have multiple studies. This can be numerous studies or different phases. Say for example, you have few studies from phase 1, a few from phase 2, and a couple from phase 3. And when you are trying to pull all these studies together into a very harmonious data set which can be used for modeling, it can pose some tedious challenges, and you need to make sure you have your imputation rules in place which can be carried forward through all your different studies. The other scenario could be multiple dosing. So if you have missed doses, or if you're dealing with dosing diaries, then it can be a very complex task to have a very well defined dosing history. Usually this kind of scenario you see in studies which have been conducted or are being carried forward for a long period of time, where it's almost impossible to have an accurate dosing history for each individual. So again, you need to be aware of such scenarios and have your imputation rules in place, keeping in mind all these different scenarios. The other example of complex data is covariates. So covariates can be of different types, time varying, baseline, or if you have missed covariates. And the one of the most complex or messy data related to covariates can be seen if they are being captured in different units. So say for example, if your study is multi-center study and you have one part of the study being conducted in a part of a world where they capture for example weight in kilograms, and then you have the other part of the clinical study which is being carried in a different part of the world where they capture weight in pounds. So you need to make sure all the conversions are taken into consideration before you put your data set input your data set into the software for analysis. And all of this, as you can imagine, can be very tedious and very complex to handle. Then some other examples are your subjects enrolled in those clinical studies. It could be patients, healthy population, or your special population. And then the other very complex area is your comeds. So say for example, in phase 3, usually the patients that you are enrolling in your clinical trial are not only on the your test drug, they usually have the other co-meds given to them. So if you want to analyze the interaction between those, it can be very tricky to handle such situations. So please be mindful of these some of the examples that I've mentioned while you create your analysis data set. So as you can see, the data set is a very critical part of your modeling, and you need to have discussions around those before modeling begins, or if you're thinking of carrying forward with your PKPD analysis. And these data set discussions should be had with, if you have a dedicated data management team, then the scientist and the data analyst, they need to sit together, understand the clinical study, understand the data that they are dealing with, and also communicate the modeling goals, because that usually determines what kind of data you need in your input you need for to perform those analysis. And if you are going to be creating your own data set, then I highly suggest that you spend some time understanding your data, exploring the data, and make sure you have all the necessary and valuable variables that you need to do your analysis. And that's because your model is informed by the data that you feed in, and your model is only as good as your data. With this brief introduction, I hand over to Rebecca, who's going to show you an example of PKPD analysis dataset and is also going to talk about dosing data in detail.
Thank you, Mitali, and thank you, Sami, for that introduction. So the first thing I want to show you is an example of a PKPD analysis data set. As you see, it can be quite complicated. This example is representing separate records for doses, PK concentrations, and PK/PD endpoint measures. The variable DVID is your record identifier. The value of 0 are dose records. The value of 1 are the PK concentration records. And the values of 2 is the PD measure. And in this case, this is representing hemoglobin. An analysis data set can get quite lengthy. It can also include covariates, as Mitali discussed, and those would be added on as additional columns, and information would be carried across your subject's records. One thing I want to for you to notice in this example is that this first dose record, there is a date and a time for it. And this time variable, this relative time since first dose variable, is at time 0. And all of the records for that subject are ordered in relation to that time 0 first dose record. The example that I'm showing are an example of the new PPK guidance that was released last week by CDISC. As I continue to talk, I'm going to be referring to some SDTM data sets. CDISC has several formats and guidance for sharing clinical data. One is the study tabulation model, SDTM, and another is the analysis data model, or ADAM. As I show the examples today, I am speaking from my experience as an end user of CDISC data, and this is not intended to be instruction on how to create compliant CDISC data. My focus today is going to be on dosing data. Dosing data can be found in a variety of a few different SDTM domains. The main domains for dosing data where dose date and time as well as amount can be recorded is EC for exposures collected, or EX for exposures. And additional dose times may be recorded along with the PK samples in the PC domain for the pharmacokinetics concentration. Typically, dose amount is not recorded in the PC domain. There are other supplemental domains that may accompany these as well. So the supplemental domains, a supplemental domain to EC would be SUP, SUPP, EC, and there may be SUP EX or SUP PC. So if you are trying to locate your clinical data in SDTM data sets, hopefully this is helpful. Sorry. So important dosing information to feed into an analysis data set. The first most important is the dose amount. In order to create a successful model, the amount of dose administered needs to be collected and recorded, as well as the units that is being used for that value. I am just differentiating between collecting data and recording data. Data may be collected in a CRF, but it also needs to be recorded in the data sets that are shared with the end users. In my examples, the recorded data sets are the SDTM data sets. Another piece of information that's important to record for dosing information is the first dose date and time. As the previous example showed, prior to analysis, your analytical data set is time ordered in relation to that first dose. In lieu of that, you might have a PK/PD combined data set. Some software do not like negative times. So time since first event may be used in that case, and then the records would be ordered from that first PD measure. The order of events or records in the analysis data set do inform the model. So it is important to keep track of how the data is going to be arranged prior to the analysis. So I mentioned that there are several SDTM domains where dose times could be recorded. There's both an EC or EX as well as PC. The example I'm showing here is an EC domain, and you will see these variables of EC ST start date time and EC end date time. Typically, these are shown with only dates in some of the guidance materials, but I wanted to point out that they can handle clock times as well. Looking at this example, this is one subject, and you can see that each record has a start and an end date, and those start and end date times are equivalent. So each record is representing one dose, with the exception of the record from on days 9 and 10. The start date would be day 9 and the end date would be day 10. What this is also telling me is that for each record, one tablet or one capsule was administered. I can also see that the subject was on treatment 1 from days 1 to 3 and treatment 2 on days 9 to 11, and that they were administered 2 pieces of medication, 1 tablet and 1 capsule as the treatment. In comparison, this would be the EX data set that would go along with that EC data set. So in the EX data set, you'll see that the treatment 1 and treatment 2 have been assigned a dose in milligrams. And we can also see that where it was bottle 1 and bottle 2 before, bottle 1 is your study drug, and then bottle 2 was the planned placebo in a capsule form. You see that the start and end dates translate over exactly the same as it was in EX. And the treatment 1 and 2 are also represented, but where the dose unit in EC was tablet and capsule, that translates to the EX dose formulation in EX as tablet and capsule. And the frequency is QD, or once a day. PC also has a variable for capturing relative dose, date, and time relative to the PK sample collection. Keep in mind that this is only going to capture information around a PK sample. So in this example, there was a full profile captured on day 1 of pharmacokinetic concentration sampling, and these would be the individual date and times for the PK samples. But then in this PCRFT DTC, that is where a relative dose, date, and time can be captured. So in this example, the 1 dose date time of July 1st at 7:30 is associated with the first few records on day 1. And then for the PK sample collected on day 9, you'll see another dose, date, and time captured on July 9th. So sometimes EX can be oversimplified and granular information such as the time of collection can be lost. This is a typical example and is perfectly acceptable to represent your dosing data that was collected. So this tells me that for this subject, from days 1 to 3, the subject got 10 milligram tablet and a 0 milligram capsule. And then on days 9 through 11, they switched to treatment 2 on 20 milligrams and the placebo capsule. But you can see that the dates and times, the times of collection were not captured in EX in this example. Let's assume that the prior PC data set goes with this example, and this is what you got. So you got a date range in EX, and the only times that you have captured are the times associated to the PK concentrations. In that case, those times from the PK concentration would be merged to the individual dose records on that date, and the date range from EX would be expanded to create these individual dose records. So it's going to, you would create an individual dose record on day 1, 2, and 3 for your analysis data set, and then on day 9, 10, and 11 to correlate with that window. And then the times from PC would be merged by date. For the records that did not have a time recorded, that time available on day 9 would be carried backwards and then forward. One may wonder if minutes can matter. This is what that data set would look like utilizing all the times that were captured. And minutes can matter depending on where the samples are taking in relation to the dose. One useful piece of information can be to calculate the time since previous dose. And that information can be useful to review your analysis data in plots and in general, just to review the quality of your data build. Here's an example of such a plot. By calculating the time since previous dose, you can put multiple profiles, being that there was a full profile collected on day 1 and day 7 for each individual. In this example, each plot represents one individual and 2 profiles, and you can see the differences in the concentrations visually between day 1 and day 7. I want to demonstrate the impact on an analysis data set if that first dose date and time was not collected and recorded. This is the example I first showed you. It has both the dose records, the PK records, and the PD records. Now imagine if PK was not collected on day 1 and that first dose date and time was not collected. How would that impact the analysis data set? What would happen is if only the dose date and times were collected around the day 7 dose, then these would be the only times in green that were actual times. This first actual time would then be carried backwards onto the individual dose records created back to the first day. And then the relative time since first dose would be calculated and the data set would be reordered by that relative time. By reordering the data set, you can see that this pre-dose sample for the hemoglobin then gets sorted after the first dose, which would not be an accurate representation of the actual collection of the data. Such anomalies would need to be reviewed and discussed with your analyst, and then a solution would need to be discussed and documented as well. If times are going to be adjusted to have your analysis data set represent the actual data, then it's preferable to adjust the imputed times and not the actual times. In this case, this imputed time of the first dose could be adjusted so that the first dose did get sorted after the pre-dose hemoglobin sample. Another piece of important information is to collect accurate dose amounts. In this example, this is an EX data set to where the EX dose is typically understood to be the dose administered during these date range by this frequency. So this is not a complete record of all these subjects' dose records, as it starts on day 449. And we can see that they've been on this comparator drug every 2 weeks at 20 micrograms for about a year. And then the next record, even though the EX dose says 60, once you read the information in the EX dose frequency, you can see that they actually received 40 milligrams every 2 weeks and then 20 milligrams every 2 weeks. So the dosing actually fluctuated during this time period. And likewise, they went to 40 milligrams every 2 weeks for another month, and then 100 micrograms every 4 weeks for another month and a half. And then we got creative again with how the dose amount was recorded and the contradictory information in dose frequency. By summarizing the dose administered over this time period as the EX dose, it's easily misused. This dose amount could be easily misused when creating an analysis data set and creating those individual dosing records. This is how that data would be expanded into individual dosing records. So this example retains the start and stop dates as presented in EX. And then this date, you can see the individual dose records and then the associated actual dose amounts, considering the information in EX dose freak that was provided. All of this was discussed with the data provider, and the solution was well documented and agreed upon. In this example, you also see a time from previous dose calculated. And this is an easy calculation to do in order to assist the creation of individual doses. One of the challenges with expanding doses within a date range is you may end up creating overlapping doses between these date ranges. And by calculating the time from previous dose, it's an easy indicator to identify where there might have been an extra dose created that was too close to another dose. Again, you would discuss your plan for creating accurate dosing with the analyst or the client and agree on the solution to use. Another challenging dosing data can be BID dosing or dosing twice a day. In this example, this is an EX data set that is representing BID dosing. This is for a single subject. They're getting 300 milligram twice a day, but I see that these ranges, these records seem to be duplicated, but what they are representing is an AM dose and a PM dose. And this fourth record represents a skipped dose. So on 6/17, they had a dose of 300, but then they skipped a dose as well on that day. So from this, it looks like the skipped dose was the PM dose. Along with this data set, this EX data set, they provided a supplemental EX data set. To utilize the supplemental EX dataset, you need to understand what records in the supplemental EX dataset go with what records in the EX. And I had pointed out that it was the fourth record that had the skipped dose. So this is the information in the supplemental EX dataset that goes with that fourth record. And in EX, they are capturing the time of the start date and the time of the stop date. And in EX, there's contradictory information that they were both AM times. Likewise, there was additional dose dates and times recorded in the supplemental PC file. So in the supplemental PC file, at the time of PK sampling, there was the dose date and time prior to the pre-dose sample collected and recorded, and there was the dose date time after the pre-dose PK sample collected and recorded. So you can see on 6/17, they had a dose at 12:05. So we now know that one of the doses were skipped, but one of the doses was administered at 12:05 on that day. So the solution was discussed and documented, and this is the resulting analysis data set. The dosing records here in blue are the actual times that were recorded with the SUPA-PC data. The records without amount are your concentration records, and the records with amount are your dose records. So those individual twice-a-day dosing records were created and included in the analysis data set. And you can see that on 6/17, only one dose is represented, where there are the two doses, the BID dosing on the other days. As far as time for these created dose records, you're going to see that they were created with times of 1500 and 0300. And that time came from the first recorded pre-dose sample or dose for that subject. So this first recorded pre-dose time that was recorded in the supplemental PC was utilized to impute the times every 12 hours backwards to that first dose. Again, my example showed a skip dose on 6/21, and you can see that there's only the one dose with the appropriate AM or PM time. The next example I want to discuss is IV dosing. There's several pieces of information that are used in PK/PD modeling for IV dosing. Not only is it the accurate dose amount administered, but for IV dosing, you need to know the start and the stop time of the infusion. So this is an example of an EX data set for IV dosing. It's representing 3 subjects with a single dose. It's representing, in this case, the start date time and the end date time are representative of the infusion start and stop time. And the EX dose is the volume, and we have a little bit more information about what treatment each subject was on. This protocol specified that the infusion time was 60 minutes plus or minus 5 minutes. And from a quick look, we can see that this first subject had a bit wider start and stop time that extended that 5-minute window. Along with the EX data set, a supplemental EX data set was provided. In the supplemental EX data set, information about infusion interruption was provided. So you can see that for the first subject, there was an infusion interruption, but for the second 2, there was no interruption, represented by these no flags. As well as understanding if it was interrupted. If it was interrupted, they collected and recorded a restart time as well as the restart stop time. They recorded if the infusion was complete and as well as the reason for the interruption. What I'm not, you'll notice that this stop time does agree with the end time in the EX data set, but the restart time, the start time in the EX data set was 10 o'clock and the restart time of the infusion was 10:12. From the data that was provided, we really don't know what happened between 10 o'clock and 10:12, if any dose was successfully administered or not. Other pieces of information in the supplemental EX data set was a dose level and a volume infused. But you'll also notice that with these values, there are no units specified. So you can utilize the protocol to try to understand how the dose was administered and how what these dose levels of 6, 1, and 20 mean. In the protocol, it was clearly stated that subjects were randomized to a 1 milligram per kilogram dose, a 6 milligram per kilogram, or a 20 milligram per kilogram treatment. So it wasn't clear to me where that dose amount in EX. In this table, this dose amount were the provided values from the EX dose. If one were to calculate a treatment based on the weight, because if the treatment is 6 milligram per kilogram and we know the weight, we can calculate the dose in milligrams. And to better understand the data, I was trying to figure out where this amount in EX came from. So I performed this calculation compared to the values. And as you can see, one didn't quite match. So with the discussion with the analyst, you might want to explore your data a little bit further and see how widespread this issue may be. So by programmatically calculating the dose from treatment and weight and comparing it to the dose in EX dose, you can see that there was only 14 of 60 recorded doses that did not match. Under the guidance of the analyst, if you wanted to explore this further, you could look at differences, but this would definitely be discussed and a solution would be reached and documented and implemented. So this is what a IV dose record might look like in an analysis data set. You have both the start time of the infusion dose and the stop time of the infusion dose. A rate may be calculated or it may be stationary depending on your model. A duration may be calculated, and then the amount, and then the weight. For this first record, because it was unknown what happened between 10 and 10:12, the decision was made to set the duration to the default as per protocol time of 1 hour. And a duration imputation flag was added to the data set so that that piece of information could be found at a later time. When creating analysis data sets, if you have a separate dedicated programmer from the analyst and the data provider, communication is key to creating successful PK/PD analysis data sets. The responsibility of the programmer is to communicate any data issues with the rest of the team and to agree upon the solutions with the analyst or the client. The responsibility of the analyst is to clearly communicate the dataset needs for the anticipated model if there is a separate programmer. And the responsibility of the data provider is to communicate known issues and challenges in the data that is being provided. It's also important to understand if you're working with interim or locked data. Working with interim data has its own set of challenges, and rules for working with that data should be discussed and solutions agreed upon. Another form of communication is to provide data specifications with the data. This helps the end user utilize that data. So if you're providing SDTM data, it's always useful to have a data specification included with that data. Data specification might have helped understand how that EX dose was calculated in the IV example. The protocol is key to understanding your clinical data. It helps you understand the study design as well as the treatment planned. It helps you check for expected dosing regimens and expected planned sampling. Another key piece of information to share with the person creating an analysis data set would be an annotated CRF. It can be helpful to understand how the data was collected to see if there's, and that correlation between how it was collected and how it's recorded. Having an annotated CRF would have explained how that volume for the IV was collected. Typically, the units might have been on the CRF, but not necessarily captured and not necessarily recorded in the data sets provided. It can also help explain the planned timing of the samples versus the dose, the PK samples versus the PD samples, and what to expect. As issues are discussed and solutions are created, it's important to document all of these discussions and solutions in an easily accessible place for the end user to access. Any supporting queries or output that was used in making this decision is also important to keep and document. Data specifications for the analysis data set should be clear enough to create a summary for the end user of the analysis data set. If code is going to be shared or reused at a later time, it's a good idea to keep comments in your code to assist in understanding of that code and the creation of the data set. An example of documenting the decisions and the issues with the BID might be as follows. The direction is to expand the EX start and stop dates into individual dosing records. Utilize the SUP-EX to identify the AM/PM start times for those date ranges. Utilize the dose time from the SUP-PC. And it was important to note that the protocol had the patients hold the AM doses, so that can help to the understanding of your data. The dose times from sub-PC were retained backwards and forwards in 12-hour intervals for the BID dosing. The decision to include the ACT dose flag to identify the records that had actual dates and times provided. And then it was documented that skipped doses were removed. An example of documenting the irregular dosing frequencies in the example presented was that exceptions were shared and reviewed together. There was actually an output created. That output was shared and it was documented right in the output. The decisions that were made, and that output is stored with the programs that created the analysis data set. As promised, we were going to provide some tips and tricks for checking your data. As I code, I like to keep an important piece of information to keep track of as you build your analysis data set is the number of subjects in your analysis data set, as well as the number of records compared to the data that was received. There's a variety of checks that can be performed to check for common date errors. The calculation of the time since previous dose can be calculated and then compared to planned time points. This is an easy way to identify an AM PM time that might have been mixed up, a date that did not get incremented for a full profile that exceeded past midnight. For example, if the beginning of your profile PK samples were collected on May 15th, but that last 24-hour sample was collected after midnight, it's typical to see that the date did not get incremented from May 15th to May 16th. And looking at that 24-hour sample, it would have a lower time since previous dose, because it would look like it occurred 24 hours earlier before the dose. Another typical date error is the year not being incremented. You can check the calculated dose amounts compared to the actual treatment like we did for IV. I tend to like to review example subjects and not a few rows. It's important to understand how information is being carried across a subject, and only looking at a few rows, you don't get a good sense of that. You want to check for duplicate records by subject to time and record type. Some software do not allow for duplicate records for the same record type. If there are duplicates found, again, that should be discussed and documented as to how that's going to be handled. You want to check that every subject has a dose record, and no subjects only have dose records. You want to check that time between doses compared to your dosing regimen to make sure that you didn't create any additional doses or have too few doses. You can also check covariates. You can check categorical values by looking at a summary of the min-max of that value by the categorical variable. When you're combining multiple studies, as Mitali mentioned, units may be mixed and from one study to another. And if you just look at some summaries across the studies of those variables, differences can be easily identified. It may also be useful to have some self-documenting variables within the data set so that so it's very clear to an end user what numerical values represent. In this example, the age, there's an age and an age category. If the age category is numerical, to include an age category decode variable of age cat C, it would be very clear to the end user what those limits and ranges for each category were. There might be additional data in an analysis data set that's not included in the model, but is useful to have in the analysis data set for graphical exploration or to assist in the review of the data set. In conclusion, as I hope we showed today, dosing data can be very complex and is vital to a successful POP-PK model. It's of equal importance is capturing useful information, recording it in shared data, and utilizing it properly in the analysis dataset to inform the model. Awareness is key. What data is available and what the model goal is and how the data will inform the model. And as always, keep in mind the goal of having reproducible results. Thank you for attending.
All right, I think we can now open the floor for live Q&A, Sami.
Yeah, absolutely. Thank you, Jasmine. So, excuse me. So for one of the questions is for IV data collection and the start and the stop time, do you do the times of priming the infusion set before the drug infusion? And then after the time of saline flushing of the infusion set, or do we wait for data analysis focus only on the drug infusion itself?
Mitali, I would appreciate your input here. I think I know the answer, but I would appreciate your expertise.
Yep. So I think the flushing time is usually not considered. It's the start of when the drug started infusing, and that's the time you usually consider for your analysis.
Thank you, Mitali. Rebecca, any comments? Or should we move on to the next one?
Nope, that's it. That is what I would have done with the IV dosing data, yes.
Okay. A scientist is asking, what does AFRLT stand for?
It's the variable in the ADPPK data set that is the actual time from relative to the first dose.
So is this a column name recognized by non-MAM, or do you need to rename the column to time in U control stream?
If you were to use that in your analysis data set, in your ADPPK analysis data set, you would then rename it for input into your modeling software as the time variable, or identify it as the time variable in Monolix.
Got it. Mitali, anything to add?
I think she answered it pretty nicely. So if whatever time variable you want to use as your time variable, just rename it to time. If you're using non-mem, the question is specifically for non-mem. So yeah, you just need to relabel your variable to time, and then you should be able to perform your analysis.
Got it. Jasmine, do we have time for additional questions or?
Yeah, we have a couple more minutes.
Okay, thank you. So one of the question is all the changes need to be recorded or, sorry, excuse me, I'll repeat. Is all the changes needs to be recorded, and is it usually done in R or Python and not in Excel?
That's an excellent question. So because our goal is to create reproducible results, you would want to make those changes programmatically and not on the fly. Any software can be used, SAS, R, Python. The industry is going to a more open source programming language, and those programs should be stored. And again, the goal is to create reproducible results. So anybody should be able to use that source data and get to that analysis data set in the same fashion.
Thank you.
And I'll just add that sometimes it's good to have the history for audit purposes too. So, yep.
Thank you. One more question. In addition to the graphical observation of plotted data, after imputation of missed time data, can we perform sensitivity analysis later with and without the generated data to see if the imputation has any effects on the results?
So Mitali, I need your expertise here. You can't like remove the dosing record, but there is a way to consider that imputation flag in the modeling.
Yes, that's correct. You want to have the imputation flag so that you are aware where the dose was missed or what happened there. And then you can use that for you can use different type of plotting techniques to get an answer or awareness as to where is that lying and how is that impacting or how will it impact your results.
Thank you. One more question is, do you use R for fixing data issues? What is your preferred software? I'll take a first stab at it because Lixoft, which is a France-based pharmacometrics software company, it is a part of Simulations Plus family. And yes, Monolix is one of the important software which we use for our own modeling purposes as well. But yeah, over to you, Rebecca. And Mitali, if you have any views on the question, do you use R for fixing data issues?
So yes, typically in the past, in my first 12, 13 years, I was a SAS programmer, and primarily we used SAS to help create analysis data sets. And in the last year and a half, I've switched to using R to creating those analysis data sets. And our team has a mix of R and SAS skills if the data is going to be manipulated outside of the analysis software.
Sorry, I was on mute. Thank you very much. One important question which we tried to address during our call, during the presentation as well, but maybe we could be a little bit more explicit. How do we handle the missing and inappropriate data? That is one question.
So that all depends on what is missing and what is inappropriate. So it has to be weighed with how important is that going to be to the model and the modeling goal. If, for example, you're missing all of your PK sampling times, if it's sparse sampling, that might have lesser impact than if it's full profile sampling. And it all depends on, again, how is it going to inform the model? Any missing information should be fully discussed with the team members and documented as to what the solutions are.
Thank you, Rebecca. Jasmine, I think this is all the time we have for answering questions, but attendees kindly do note that we have your questions, list of questions, and we'll follow up with you individually and answer your questions. So apologies, but thank you for sparing time, everyone. Over to you, Jasmine, for the final comments.
Yeah, thank you all. For any questions, like Sami said, we didn't get to today, we'll reach out with answers after the webinar. You can visit us on our website. I put a couple links in the chat. This webinar has been recorded for playback and will be available on our website and YouTube channel. Please allow like 24, 48 hours for it to be up. Make sure to share your thoughts in the post-webinar survey that follows. This concludes our webinar for today. Thank you, everyone.
Thank you.
Thank you.